Again. So I'm here to introduce our next speaker, and Professor Teng from Department of Chemical and Medical Engineering. He's a professor chair in the University of Florida and Florida State University College of Engineering.
Thank the organizers, Joe, JK, and for the invitation. I truly enjoyed the talks, and also for all the speakers I have. Fascinating so far. And we have been working on human mesenchymal stem cells for bioengineering and making use of these cells, how to make best of the MSCs for therapeutic application. MSC is one of the most commonly used stem cell in therapy, and among this large number of trials, about 11% are in cardiovascular disease, including stroke. And among these cells, you can clearly see that many of these trials, majority of these trials, also use culture-expanded MSCs, and because of the clinical dose required. And so far, there are about over 40 heart failure trials using MSC and about 20 in stroke. And these cells are also from different sources, from adult source and also fetal source. And even though adult source, the MSC, are most commonly used, fetal source MSC actually are being more tested and have demonstrated safety and efficacy. And each type of MSC has their own kind of properties, and the fetal source are highly proliferative, but adult source has higher expression of immune suppressive property and also highly more pro-angiogenic. The mechanism at the beginning of MSC research are people think this cell can differentiate, and now people probably appreciate more of their trophic factors, their immune suppression, and also the pro-angiogenic and the pro-survival effect. To achieve this goal, the MSCs must be properly prepared. So it has to be properly equipped with functionally equipped and also survive long enough so that you can deliver the factors as a meaningful dose. However, even though the safety is established, the MSC has been consistent results has not been demonstrated. One of the barriers people have talked about and also alluded to this morning and through today is how to deliver enough MSCs and how to preserve their therapeutic potency. So the challenge really is how to produce this functionally equipped MSC at a clinical scale and how to enhance their survival after transplantation. And after transplantation, where these cells go and how to track their fate, and finally is the sourcing. So it's adult or fetal sourced MSC. In engineering, actually, the one challenge is underappreciated is MSC requires a large-scale expansion, but the current technology really is not designed for expanding these cells, adherent cells, in larger scale. If you think about the MSC are isolated from a very well-organized, well-controlled, regulated in vivo environment, put them in an in vitro environment, and then expand them many fold, put in the human body, they really undergo a lot of changes. Actually, looking back from a couple of years, materials research have demonstrated these cells respond to many different cues, being biophysical, biochemical, and also the topographical and the geometrical cues. So they do accumulate a lot of this information during their short span in vitro. And the study have shown consistently rapid declining CFU5 forming capacity, increasing senescence, and also enlarge of cell size. So these cells, once you expand them, they become much larger compared to the innate cells. So our goal has been how to manufacture this type of cells, therapeutically competent cells, and at a sufficient scale as an off-the-shelf product. And the one approach we are taking is how to make these cells into 3D aggregates. And aggregation is not a new phenomenon and has been observed in many different biological processes, mainly in embryogenesis, morphogenesis, and also angiogenesis. And also in vitro, people have been using aggregation to isolate progenitor cells, cardiac progenitor cells, epithelial progenitor cells, and neuron progenitor cells. In MSC, in for MSC, were originally isolated based on plastic adherence. So it's counterintuitive to see these cells actually has spontaneously organized into 3D aggregates. And this slide shows that if you add, for control MSC in about overnight, 12 hours, they aggregate into a very well-defined aggregate. But if you block EDTA, use EDTA to block cell adhesion and use cytokeratin D to break up the F-actin, you actually completely stop them from aggregating. And what's the impact of aggregation? The first impact of aggregation is significantly reduce the cell size, and then making cells much circular, smaller size. And the aggregate-derived MSC also has a significantly higher CXCR4 expression and also migration towards cytokines. So that's first one. And also aggregation revert this back revert these cells back to their pluripotent state with a significant increase in stem cell genes and also the proliferation. So these cells also preserve this property after several passages. You dissociate this cell, put them back in planar culture. These cells still maintain this property for a couple of lineage. They also have a much better survival. So these are the genes induced by aggregation. So PI3K expression as well as the stress-induced genes are significantly upregulated. If you look at their resistance to ischemic stress, the aggregate-derived cells, both aggregate and aggregate-derived cells, have higher survival under ischemic stress. And also, finally, aggregation also enhances secretion of trophic factors. VEGF, HGF, and these major factors, PEG2 and other factors are significantly upregulated when cells are aggregated into 3D aggregates. And also their anti-inflammation response is much higher. And although this phenomenon is size dependent. So what's the mechanism? For a long, long time, people think that aggregation is because of inducing of hypoxia zone inside these aggregates. But actually, by measuring oxygen tension, we will see that the reduction of oxygen tension is actually limited for the aggregate size up to 50 K per aggregate. However, the mitochondrial activity is significantly reduced. So for large aggregates, the majority of the aggregates actually operate within the reaction limited zone until the size increase up to 30 K. And, but because of the mitochondria is reduced, that's why the oxygen consumption rate is induced. The reason for this mitochondrial inhibition is believed to be because of the compaction itself, really not about diffusion limitation. So in summary, basically, the mechanism we believe undergoing for regulating this cell aggregation-mediated mechanism is the cells are changing from a mixed glycolytic and oxidative phosphorylation phenotype to a predominantly glycolytic dominated phenotype. Because of the increase in glycolysis, the secretion of trophic factor is significantly upregulated. And also because of mitochondrial defect in this 3D aggregates, the activation of PI3K is upregulated, so cell survival is increased. So the next step is, this is good strategy to enhance the cells. Can we make these aggregates in larger scale? And as an engineer, what we are concerned is, can we deliver this technology to application? And there are many reactors people have tested for growing aggregates of different sorts, different types. And many of them are designed to grow single aggregates. What we want to test is actually use a new technology. This is many people of you probably familiar. This is a low shear GE wave bioreactor that generate wave motion and at a very low shear stress. And we have studied that actually this shear, by regulating the shear distribution inside this fluid flow plates, you can actually regulate the size distribution of these aggregates, and you can achieve relatively uniform size distribution. Exploiting this technology, we can also look at assembly of cell of different types by making different aggregates. And this one has a couple of smaller ones first formed, under one flow condition, and then add another type of cells, and these cells, they assemble into this three-dimensional structure. By changing the geometry of this, you can organize the cells into different shape. Another one is, a recent paper is we developed a microcarrier that surfaces are coated with thermoresponsive plastic, and then first the cells will be expanded on this microcarrier, and then by reducing temperature, the cell will detach as cell sheets, and then under certain hydrodynamic conditions, they reorganize into this well-defined aggregates. Finally, I want to show some of our recent work in stroke, and we don't have any heart-related in vivo study, but I want to use, hopefully next year, we can find a collaborator to work on that. And in this study, we basically injected both aggregate-derived single cells and also aggregates in stroke injury. So first, we form these MSCs, and then for 2 days as aggregates, and then dissociated them into single cells, and then label them with SPILE nanoparticles, and then inject into a ischemic brain stroke model. And these cells, we did histology and can clearly show the cells are still present in the aggregates, in the brain. So this is an MRI segmentation of the brain injury. You can see once you inject this MSC in brain, and these cells migrate to the injury site. And we also compared cells from planar culture and also three-dimensional aggregate-derived MSCs. And the percentage of lesion reduction is significantly higher in the aggregate-derived cells. So these cells survive better by MRI measurement. And we also performed, taking advantage of the facility we have at National High Magnetic Field Lab, we also measured the sodium expression, the sodium amount in the stroke lesion. And from the proton T2 image, we didn't see any significant difference between these two different cell types. But from sodium, we can clearly see increased depression, increased recovery of lesion site for the aggregate-derived cells. Finally, I want to show some of our recent work on direct injecting of these cells as three-dimensional aggregates in the stroke lesion. These are two clusters of aggregates. For each animal, we receive around 20 aggregates. And one of the aggregates is located in the upper ventricle, and this one is lower ventricle. And you can see by 7 days that the cells start to migrate toward stroke site, and this is the stroke lesion site. The segmentation picture shows more clearly that the aggregates can maintain their integrity inside this brain environment for up to 7 days, and by day 7 you will see that the aggregates that have access to white tract actually start to migrating towards the stroke lesion site. And this tells us that the aggregate can be maintained viable in the injury site, and also they basically potentiate a broader set of spectrum of properties by this cell-mediated aggregation. And also, both aggregate and aggregate-derived cells maintain this property. So you can use either the aggregate-derived cells or aggregate directly. So we wanted to, and finally we have shown that actually injecting, producing, these aggregates can be produced in the reactor system with defined hydrodynamic conditions. And finally, I want to acknowledge the students who performed this work, and Yijun is the main reason. The first two are, and also Angren are two recent graduates. And also the collaborators, a long-time collaborator with Bruce Bonnell at Tulane University on MSCs, and also Dr. Grant and Rosenberry at the National High Magnetic Field Lab. And thank you for your attention. Yeah.
Thanks for the great talk. We have time for questions? Any questions?